The response at the lecture next Friday will be in the same format, same platform, or we are going to use Google Meet? No, can't hear you, Raswaiza.
Lecture with postgraduate, Prof.
Yes, yes, yes.
It will be using the Gmeet.
Gmeet. All right.
Yeah, because I think there are less than 100 of them.
I see. Okay.
Yeah. We wait for a couple of minutes, Prof.
Yeah, no problems. No
Yeah.
problems.
Assalamualaikum warahmatullahi wabarakatuh and a very good afternoon. I would like to welcome everybody to our TRANSCENT webinar. The title is Research in Leptin. Before we start, I would like to introduce our speaker, Prof Dr. Harpinder Singh. So Prof Harpinder is a professor of physiology at the Faculty of Medicine, University Technology Mara. He joined UiTM in 2007 after serving at the Department of Physiology, School of Medical Sciences, University Science Malaysia from 1985 to 2007. He obtained his PhD in 1985 from Manchester, United Kingdom. Over the years, his research interests have included topics in renal physiology, calcium metabolism, exercise physiology, hypertensive disorders or pregnancy, glucose metabolism, high blood pressure, and on endothelial cells and adipocytes in culture. Recently, he's also have massive research in leptin in physiology. Prof is a recipient of a number of research grants and he has supervised numerous postgraduate students over the years, and he has over 80 publications in peer-reviewed journals. So without further delay, I would like to invite Prof Harpinder to deliver his talk.
Thank you very much, Rosfaiza. Can you hear me? Can you all hear me? Yeah.
Yes, Prof.
Thank you. Yeah. Good afternoon, everybody. Yeah. First of all, I would like to thank the organizers for inviting me to talk about my research work from the last few years. Now, what I thought I'd like to share with yourselves this afternoon is our work on leptin, which we have been doing over the last 20 years or so. Now, in the process, I hope you will notice how it all began and how it has now grown over the years. And more importantly, I hope it will help generate more new or novel and interesting areas of research, particularly the role of leptin in disease. Now, in the first few slides, about 9 to 10 slides, I shall introduce leptin just for the benefit of those who may not be very familiar with it. And then after that, I shall go on to talk about my research, about our findings that support the role of leptin in disease, especially in obesity-related diseases. Can you see my slides?
Yes, Prof.
Yeah, well, so how do I move my slides? Because I can't see my own slides here.
Wait, Prof. All right. Can you all see the slides now? Yeah, Prof.
Yeah.
Now, yeah.
Right. All right. What is leptin? In fact, leptin is an adipocyte hormone, and it's a product of the OB or the LEP gene. And leptin was first identified in 1994 after almost a 50-year search for a factor that helps control appetite and body weight. It is a 16 kilodalton protein, and it consists of 167 amino acids, and it's known to derive its name from the Greek word called leptos, which means thin, something that makes you thin. And it has a tertiary structure that is very similar to that of cytokines. And of course, the relevance of leptin in humans became evident sometime in 1997. And this was following the first report of a case of human congenital leptin deficiency that caused severe obesity in a child. And since then, a number of human cases of leptin deficiency have been reported. There are some 8 to 12, I think, some 8 to 12 cases so far. And in humans, the leptin gene is localized on chromosome 7, and it spans about 18 kilobase, consisting of 3 exons separated by 2 introns. Leptin, for most part, is or mainly synthesized by the white adipose tissue. But we know today that small amounts of leptin have been detected in the fundus of the epithelium of the stomach. It is also produced by some cells in the intestine. It's produced by the syncytial trophoblasts of the placenta. Small amounts are also produced in skeletal muscle, mammary epithelium, and also the brain. And it has a secretory pattern that has a diurnal rhythm, where you have higher rates during the evening as you approach towards the evening, peaking in the middle of the night, and then it declines again to low rates in the morning. Now, these might have something to do with our dietary habits as well and our lifestyles. And interestingly, there is a sexual dimorphism as well. In other words, there is a gender difference. And in fact, interestingly, this difference becomes evident from very young, even in the newborn. Generally, leptin levels, serum leptin levels are higher in women than in men. And leptin concentration in the CSF is also higher in women than men. And interestingly, leptin binding protein levels are lower in the blood of women than men. And therefore, the amount of free leptin circulating in the women is generally higher than in men. And in terms of secretion, the subcutaneous fat has a higher secretion rate than visceral fat. And also the size of the fat cell. The larger the fat cell, the higher or the more leptin is secreted from that cell. In terms of its action, of course, leptin binds to its receptors, and to date we have 6 different receptors have been identified, or 6 receptors have been identified. They can be broadly classified into 2 groups. You have the long form and the short form. The long form, of course, is the main form that actually is involved in the cellular actions of leptin. And the short forms perhaps are involved in the transport of leptin across cell membranes. And the soluble form, which is OBRE, that is how leptin is transported. It is bound to this receptor and transported in the blood. In terms of its mechanism of action, to date we know that leptin actually acts through 5 different pathways, depending upon the cell type. Of course, it acts in the hypothalamus, for example. Its action is mediated through the JAK-STAT pathway. It also acts through the AMPK pathway, acts through the mTOR pathway or the PI3K pathway, and also the MAPK pathway. So these are the 5 pathways that leptin is known to exert its action. Now, what are the major functions of leptin? In terms of its functions, the major function of leptin is that it actually reduces food intake and increases energy expenditure. In other words, it is involved in the regulation. It's a long-term regulator of appetite and food intake. In general, leptin reduces food intake, and this action is mediated through its effect on the hypothalamus, where a number of neuropeptides are involved, including neuropeptide Y, a Gauty-related peptide. These are downregulated by leptin. And CART and alpha MSH, these are upregulated. These decrease appetite, whereas NPY and a Gauty-related peptide increase appetite. So leptin actually suppresses NPY and AGRP and upregulates CART, which is cocaine amphetamine-related transcript, and also alpha MSH. And it also evidently increases metabolic rate by increasing sympathetic activity. In addition to that, of course, it might also have some effect through the hypothalamic pituitary thyroid axis. And there is also evidence to show that actually leptin alters the leakiness of the membranes, and this consequently uncouples some of the energy transfer, and it is lost as heat. So that's how it actually regulates metabolism. The other well-known function of leptin is in the immune system, and we know that it is necessary for normal immune function. And this has been evident for a very long time. And it is known that obese children that have leptin deficiency, they have a greater incidence of respiratory tract infection. And in fact, it has led to death in a few of them too. And family members with leptin deficiency and with early onset obesity, it seems are 25 times less likely than normal weight children to survive. And this is purely because of the infections that they get, the recurrent infections that actually lead to their death. And we also know that leptin deficient patients who had or have reduced CD4 T cell populations or hyporesponsive T cells, they also have severely reduced interferon gamma expression, reduced levels of cytokines or interleukin 10. But when there is leptin replacement or when leptin is given to them, that increases their CD4 numbers, it increases the interferon gamma expression, and also improve T cell function and decreases TGF beta to normal levels. So there is quite a bit of evidence suggesting that leptin is involved in the immune function. In general, we know that leptin modulates the innate and adaptive responses by repressing proliferation of regulatory T cells, modulating T cell and monocyte function. It also enhances phagocytosis and coordinates cytokine expression during infection. Apart from its function or its role in appetite regulation, immune function, leptin is also known to be involved in the regulation of sexual maturation and reproduction. And how do we know that? This we came to understand when it was reported that hypogonadism and infertility are some of the common features of leptin deficient mice. And we know that exogenous leptin administration then restores the reproductive capacity in some of these mice, suggesting that leptin is required or necessary for sexual maturity. And we also know that administration of leptin to prepubertal mice and non-human primates as well accelerates puberty. And there are cases of human leptin deficiency. For example, there's a case of a 34-year-old woman and 3 adolescent girls. They had no pubertal development and had primary amenorrhea. They were leptin deficient, and supplementation of leptin actually restored some of these reproductive activities. And there's also a case of a 22-year-old leptin deficient male who had hypothalamic hypogonadism. In normal children, leptin levels tend to increase before puberty, and they reach their peak at or around the onset of puberty. And there is a significant relationship between the onset of menarche, for example, in females, and serum leptin levels. And there is an inverse correlation between serum leptin levels and the age of menarche. Higher the leptin levels, lower the age of menarche. And in fact, this is one of the reasons as to why young girls actually reach menarche a lot early these days than they did about 100 years ago. Because of good diet, they tend to have more body fat, and therefore they produce more leptin. And the body physically is already ready for reproduction. They may not be matured mentally yet, but physically they are already ready for reproduction, as it were. And leptin perhaps may be the factor that is involved. In fact, leptin is a trigger for puberty. Now, the precise mechanism by which leptin triggers the onset of puberty is not clearly apparent. But we all know that when you administer leptin to leptin deficient mice, the OBOB mice, that increases the secretion of LH and FSH. And also leptin accelerates the Gnrh pulsatility when you measure these LH and FSH, not the amplitude, but just the pulsatility in the arcuate hypothalamic neurons. But very interestingly, the Gnrh neurons do not have or do not express leptin receptors. So how does leptin then stimulate the GnRH neurons and trigger puberty? Now this is a proposed scheme of events. It's possible that leptin may be doing that indirectly. We have leptin that's produced by the adipose tissues, and it perhaps binds to the ventral pre-mammillary nuclei or nucleus in the medial hypothalamus, and these neurons from these nuclei or nucleus actually have axons terminating on the GnRH neurons. So it is possible that the action of leptin on the GnRH neurons may be mediated through the ventral pre-mammillary nuclei. Now it's also possible that the action of leptin may be mediated to the kisspeptin neurons, because there are leptin receptors on the kisspeptin neurons, and there are kisspeptin receptors on the GnRH neurons as well. So it's possible that these could be the other mechanism, the other pathway. The third one could be through the hypothalamic nucleus, where through the POMC or the CART neurons as well, because both of these are supposed to also influence the release of GnRH. And lastly, it's possible that leptin might have a direct effect on the gonadotropins in the anterior pituitary to stimulate FSH and LH. So this is the current scheme that has been proposed, and in fact we have this is drawn by ourselves. And if you want to know a little bit more about leptin and the physiology of leptin, I suggest you may want to read this article that was a review article that was published by ourselves recently in our journal, the Journal of Clinical and Health Sciences. And the title is Leptin, a pleiotropic factor in physiology. Although I've only shown here about 3-4 functions of leptin, but leptin is now believed to have a lot of roles in physiology, because its receptors are distributed all over the body. So it might have a lot of permissive roles as well. In other words, you require leptin for normal physiological functions. Now, how did I get involved in leptin research? Where did it all start? Sorry. Well, it was just over 20 years ago. And it started off actually when I was doing some work on exercise physiology. And we have a situation, a problem where we have athletes who actually don't feel like eating immediately after a heavy bout of exercise. There is something what we refer to as exercise-induced suppression of appetite. And it's important actually for some athletes who are actually participating in multiple events that they eat food and replace the glycogen that has been used immediately after the event, somewhere between 2 hours completion of exercise, so that they are ready for the next event. But if they are not consuming the calories, or they don't feel like eating, they will not be able to replace the glycogen that has been used. So we just wondered, at that time there was a lot of talk about leptin. Leptin was just discovered, and we wondered if leptin was actually involved in these exercise-induced suppression of appetite. So we did a very preliminary study to look at it, but our study did not reveal any significant differences or changes in leptin before, during, or even after exercise. But of course, the question still remains. It's possible that leptin might be involved, and it could be through leptin sensitivity rather than actual changes in leptin levels. So there is something that we can look at, and if anyone is interested out there, we could discuss to see how we can actually look at the effect of exercise on leptin sensitivity. But nevertheless, that is how it all started. But at the same time, there was also, when I was in USM, there was also work going on in reproductive physiology. They were looking at some of the herbs and the effects on male reproductive function, the effects on sperm production, and so on and so forth. And we knew at that time that leptin was required for sexual maturation, And we knew that actually when leptin was given to pre-pubertal mice, it actually increased or brought forth the puberty, and also it increased the spermatogenesis in these rats. So we wondered that if we gave leptin to male rats, would it increase the sperm count in these rats? So that was our hypothesis. Remember, that's how we started off. Our hypothesis was if we gave leptin to male rats, it will increase the sperm count and whatever the sperm parameters were. So what we did was we actually submitted a short-term grant to USM, and we managed to secure a grant for that. And we got a postgraduate student who came along and did some work with us. So what we did here was we actually got these Sprague Dolly rats. They were normal weight. We did not use obese rats because we know that the adipocytes produce more than leptin. In fact, the adipocytes are now known to produce over 50 different adipokines. So we wanted to actually see the effect of leptin in isolation, as it were. So we chose normal weight rats, and we gave the rats daily injections of leptin in doses of either 5 or 10 or 30 micrograms per kilogram body weight intraperitoneally. It was given daily, and we gave it for 6 weeks. And then we euthanized the animals and did some measurements of sperm parameters. And what we found was, instead of increasing the sperm count, we found that actually those rats that were given leptin had decreased sperm count. Not only that, these rats also had increased fraction of sperm with abnormal morphology. Besides that, what we also found was that leptin also affected the testicular histology. Both the seminiferous tubular diameter and epithelial height were lower in rats that were given leptin. And it also increased the levels of FSH and LHH that we already knew from before that was one of the actions of leptin. But there was no change in the level of testosterone at this dose, at the doses that we used. And of course, we published this paper. This was the first report of its kind, by the way. There have never been reports about the effect of leptin on sperm in matured rats before, or in anybody for that matter. So this was the first report of its kind. Then I moved to UITM in 2009. I always like to include this story because it tells us something as well. We submitted a proposal for funding under the FRGS scheme, but the proposal was rejected. And the reason that was given to us by the Kementerian was that the work was not novel, and they cited this reference. They asked us to read this reference, and they used this as a reason as to why the work was not novel, and it was rejected. Now, we had actually cited this paper in our proposal in the literature review, and we had categorically stated in that review that leptin may not have any effect on mature ejaculated spermatozoa, but we hypothesized that it might have some effect on the spermatogenesis. But obviously, whoever reviewed it did not take note of that. They did not take note of what the authors of this paper had also concluded. They actually said human ejaculated mature spermatozoa. And I did not think that it would act or have any effect on the matured spermatozoa, although we know that there are leptin receptors found on the matured spermatozoa. But some people, we do not know what they are there for, but some people believe they may just be contaminants, probably picked up by the spermatozoa as they are coming out or something like that. So we just do not know exactly what their role is at the moment. But nevertheless, we did not give up. And that is something you must remember. Even if your proposal is rejected, you don't give up. We did not give up. What we did was, in the following year, we actually submitted the same proposal word for word, and we submitted it for the ERGS grant this time. They no longer do the ERGS grants, by the way. And fortunately for us, the grant was accepted. So what we did was we repeated that study, but this time what we did, we just used one dose of leptin. All right, we used 60 micrograms. We knew we could see the effect within 2 to 3 weeks when we used doses like 10, 15 and 30 micrograms per kilogram body weight. 5 micrograms, it took a longer time. So to be certain that we will get a good effect, we decided to do the study using 60 micrograms per kilogram body weight of leptin. And what did we find after 6 weeks? Again, we found the same thing. We found that those rats that were given leptin had lower sperm count and also a higher fraction of abnormal sperm. And around about that time, another paper appeared in the literature, and the authors from overseas, the authors virtually repeated the same study. The design of the study was virtually the same as that we used in our first publication. And they came out with the same results as well. Not only that, what they also found was that actually leptin increased the production of free radicals in that study. So that was a hint to us as well that some of the effects of leptin may be attributable to its ability to increase free radical production. In other words, it might cause oxidative stress. So in this study, what we did, we also measured the marker of oxidative stress. And this is the marker of DNA damage due to oxidative stress, which is 8-hydroxydeguanosine. And look what we found. We found that the level of this particular compound was significantly higher in rats that were treated with leptin. And it was some 7-fold higher. Nearly 7-fold higher. And to confirm this further, that there was actual DNA damage and fragmentation, what we did was we performed the COMET assay, which actually is single gel electrophoresis. And what you have is here a picture. This is of normal spermatozoa. And this is from rats that were given leptin. And you notice that there's DNA fragmentation. You can see that lots of them have tails. We quantified the COMET assay, and we found that all the parameters indicating DNA damage were significantly higher in leptin-treated rats. Now, to make sure that this effect was primarily because of oxidative stress, we did tunnel assays. And what did we find? This is of normal rats. This is of rats that were given leptin, and look at the evidence here. So it suggested that the effect of leptin on the testes was probably mediated through oxidative stress. And we also determined the level of a protein called histone in the sperm. And we know that during spermiogenesis, the histone that is present in the DNA, in the chromosome of the sperm, is actually replaced by another protein called protamine. And the purpose of this is to make the sperm more compact. And this is supposed to protect the DNA from free radical attack. And what we found was very interesting was that there was still more histone, the level of histone in the spermatozoa of rats or in the chromosome of rats that were treated with leptin was significantly higher than that in the controls. So in other words, what we understood from here was that perhaps the chromosomes or the DNA, for example, in the sperm of the rats that were given leptin was not compacted properly, and it was exposed to free radical attack. And that was the problem. That was perhaps causing the problem and causing the lower sperm count and also the morphologically abnormal sperm. Now, we hypothesized that this effect involves oxidative stress. So we asked ourselves, can we prevent this by giving antioxidants? If we were to give an antioxidant together with leptin, would it prevent the effects of leptin? What we did was here, we again repeated the study. We gave the animals leptin for 6 weeks. Along with it, we also administered melatonin in drinking water in doses of 10 milligrams per day and 20 milligrams per day to see what the effect was. And look what the effects were. This is the control animal. This is the group that was given leptin. And these are the groups that were given melatonin. And as you can see, melatonin, when given at a dose of 20 milligrams per day, actually prevented the effects of leptin completely. So it supported our hypothesis. Similarly, when you looked at the fraction of sperm with abnormal morphology, again, it was lower in rats that were given melatonin. In short, what it did was it actually melatonin prevented the effects of leptin, the adverse effects of leptin on sperm. And we measured the level of 8-hydroxyguanosine, and again we saw the same pattern. Right? Melatonin actually prevented the increase in the level of this marker of DNA damage. Similar results were evident in the tunnel assay. This is the normal. This is the leptin treated. This is the rat that was given melatonin together with leptin. Yeah. We published most of these findings in the journals recently. And we asked ourselves, the next question we asked ourselves is, which possible pathway could be responsible for the adverse effects of leptin in sperm? So I mentioned earlier that leptin, the action of leptin follows 5 pathways, depending on the cell type. And we hypothesized that since it's involving oxidative stress, energy metabolism, oxygen consumption, it's possible that the PI3K or the AMPK pathways might be involved. So we designed a study where we actually gave leptin to rats. At the same time, we gave inhibitor of the PI3K pathway or the inhibitor of the AMPK pathway. Right, so we have this dosamorphine is the inhibitor of the AMPK pathway, and we have these LY something like that, that is the inhibitor of LY294002, that is the inhibitor of the PI3K pathway. And see what happened. This is the normal control. This is the rat that is given leptin, this is the group. This is the group that was given dosamorphine, and it had no effect. In other words, it's possible that the AMPK pathway is not involved. But we saw that the PI3K pathway inhibitor actually not only prevented the effects of leptin, but also actually improved the sperm count. Similarly, also, it prevented the effects of leptin on the morphology and improved the morphology of the sperm. And if we look at the other parameters, of course, in the main, what it pointed out to us was that it is the PI3K pathway that is probably involved in this action of leptin. And we have published this work recently, suggesting that the effect may be the effect of leptin. There is no doubt in our mind that leptin has adverse effects on sperm, and these effects are most probably mediated by the PI3K pathway. Now, we are waiting for confirmation. We probably have to repeat this study, or we wait for someone else elsewhere doing this study to confirm these observations. Now, we next ask ourselves, are these effects reversible? Can we reverse these effects? We know we can prevent them. Are they reversible? Now, for this, what we did was we gave leptin to rats again for 6 weeks, and then we stopped the leptin, and then we observed them afterwards. What we did was we had a group of rats. We gave them leptin for 6 weeks. Then after that, we started euthanizing a group of rats every week for about 8 weeks afterwards, and to measure the various sperm parameters. And look at what we found. Here we have normal control. These blue bars are normal control. And here are the animals that were given leptin. Immediately upon completion of 6 weeks of leptin, that was what the sperm count was. In rats that were actually euthanized about 3 weeks afterwards, look at what the level was. It increased. 6 weeks afterwards, it was increasing, it was higher. And 8 weeks afterwards, it was near to what it was normally. In other words, the effects of leptin were reversible. The same pattern was evident in the the morphology of the sperm, it decreased as the duration progressed. In other words, the effects of leptin were not permanent. They were reversible. But the important thing was also that we observed was it actually took more than 8 weeks for the effects to disappear. And we know that a sperm cycle in a rat is about 56 days, which is about 8 weeks. So what it indicated to us was that the effects of leptin, which we had hypothesized a lot earlier when we first started this study, when our proposal was rejected, that the effect is occurring somewhere upstream and not very much on the matured sperm per se. So therefore, because the effect was somewhere upstream, so it took a longer time to wash away. And if you look at the effect of this reversal on the marker of DNA damage, 8-hydroxyguanosine, and you can see the pattern is also decreasing here. And by the time 8 weeks after the cessation of leptin administration, the levels were not very different from that in the control. We looked at the COMET assay. Again, the same pattern was evident. Now, we've published these results, and they were well received, and they've been cited many, many times now as well. We have also proposed the possible pathway that may be involved in this action of leptin on the sperm. And we hypothesize that what happens is leptin binds to these receptors, it stimulates the PI3K pathway, and it decreases the antioxidant enzyme. By the way, we also have done, I have not included the data here because it's quite a bit. We have done some microarray analysis and RT-PCR to confirm what we found that when we injected leptin, it actually caused a change in the expression of nearly 5,000 genes. Some were upregulated, some were downregulated. And there are still so many genes that we are looking at. We have to really identify what they do and what they are, the function is. But it just indicated to us the impact of leptin, what impact it has on the cells. And here we notice that it actually suppressed the expression of genes that are responsible for the antioxidant enzymes like SOD, GPX, and the catalase. The expression of these enzymes was decreased in leptin-treated rats. So from there, we assume that what it does is perhaps decreases the antioxidant enzyme concentration, and therefore it decreases the antioxidant capacity in the cells. We also know that the PI3 leptin actually does affect the mitochondria, and it is through perhaps the PI3K pathway it increases ROS production. So reduction in the antioxidant enzymes, increased ROS production collectively will induce oxidative stress. Yeah, and mentioned about the histone. There is still plenty of histone in the sperm, so the sperm are not compact, the DNA are not compact, and therefore they are more susceptible to oxidative stress or free radical attack. So that will cause DNA damage or fragmentation. And all this collectively will lead to what we see happening in the cell, that is apoptosis, and not only the sperm cells, but also in the seminiferous tubules as well, as you would have noticed from the tunnel assay. Yeah. So all these collectively together will induce apoptosis, which will actually decrease the sperm count, and also will affect the morphology of the sperm, and may also impact the sperm motility. So this is what we have proposed, and this is it has been included in some of our recent reviews, that have been published. So what is the significance of our findings? What does it tell us? Now, we know that the prevalence of infertility is higher in obese males. And we also know that sperm analysis in obese males reveal low sperm count, higher fraction of sperm with abnormal morphology. There is also evidence of increased DNA fragmentation, increased oxidative stress. All these are seen in rats, normal weight rats that are given leptin. And if you look at the literature, there have been a lot of papers published in the last 10, 15 years. And you can see a lot of study, though they have not directly related it to leptin. They have just published where they found that men with BMI of more than 35 kilograms per meter square had a lower total sperm count and considerably more sperm with DNA damage Some studies did, some of the studies did not, of course, find any difference. There was another study here that found a significant correlation between BMI and abnormal morphology. And there were also other studies where they found the incidence of oligospermia and prevalence of low progressively motile sperm count with increasing BMI. And few other studies here, but there was a review that was published in 2012, and the authors here actually reviewed 23 studies on sperm function, and they found that 15 of them reported lower sperm count concentration in obese males. So really, a lot of these epidemiological studies or these cross-sectional studies in humans do suggest that there is a very strong correlation between obesity and infertility, and more so between the sperm parameters. And of course, there is also evidence that shows that in subfertile men, obesity is 3 times more prevalent compared to male partners of couples with idiopathic or female factor infertility. And also the odds ratio for infertility is 1:20 for overweight men and 1:36 for obese men, relative to men with low normal BMI. And we also know that there are a number of hormones that are different as well between normal weight men and obese men. So if you look at the fat mass, for example, a control with a BMI of 17 and a moderately obese with a BMI of 27 and a severely obese with a BMI of 51, and you look at the testosterone levels, they're significantly lower in the obese individuals. You look at the testosterone levels, they're also lower. You look at the sex hormone binding proteins, I mean, they're relatively low. Look at the estrogen levels, they're significantly higher. And if you look at the testosterone estrogen ratio, and you will see the difference. And you see the leptin levels, they're significantly higher in obese individuals. So there is no doubt actually that, it's possible that these effects that we are seeing in the obese individuals may be related to leptin. All right. And interestingly, of course, most of these effects that you are observing or that have been reported in the obese males are evident also in normal weight rats that are given leptin. And we know that the obese males have higher serum leptin concentrations. So what we are saying is that in addition to perhaps hormonal changes, because all along what has been believed is that the higher level of infertility in obese individuals is probably due to the hormonal changes. So it probably is affecting the hypothalamus-pituitary gonadal axis, or it could be the local temperature effects. That may well be true, they may still be there, but I think there is one other factor that we are trying to say is also involved that we must not overlook. So we are saying that it is possible that apart from these 2 factors, hormonal temperature factors, the leptin, the serum leptin concentrations in obese individuals may also be contributing to the infertility. We have actually written a few reviews recently on this topic, and you may want to actually read some of them, where we have actually advanced these hypotheses and presented evidence from humans and also mostly from animals from our work, where we are proposing that leptin could be the factor, could be the link between and the reduced fertility in obese individuals, and obesity, if you like. Now, that's one area that we have been working on. I would like to just share with you a few other areas on leptin that we have been working on. And the other area that we have done some work on leptin is in the hypertensive disorders of pregnancy. I've had this interest in hypertensive disorders of pregnancy starting from my interest in calcium metabolism. There was a lot of talk in the 80s and the 90s that calcium might have some impact on blood pressure. And I just wondered that whether calcium was involved in pregnancy-induced hypertension as well, or whether the other divalent cations might be involved, like magnesium. So what we did was, this was in USA when I was still in Penang at that time in the late 1980s, early 1990s. We actually measured serum and urinary levels of these divalent cations like magnesium and calcium. And what we found was that their levels were reduced in women with pregnancy-induced hypertension. And we repeated that study subsequently in Kotabaru, and we more or less found similar results, that the serum calcium levels were significantly lower. And also the urinary, in addition to urinary divalent cations, we also found that the plasma renin activity was also low in these women with mild pregnancy-induced hypertension. And similarly, of course, there was another study that we did in diabetic pregnant women. We also found that the serum calcium and magnesium concentrations were lower. Generally, what we found was we measured the serum calcium levels and parathormone in normal pregnancy in male women, and we found that in the main, normally, serum calcium level decreases by about 4 to 5% during normal pregnancy, particularly after the second trimester and leading towards the third trimester. And that is the time also the fetus is actually trying to accrue a lot of calcium. But women who develop hypertension during pregnancy, the fall is even more. Their serum calcium level decreases by some 15%. So there may be some basis. There was at one time there was a lot of talk about calcium supplementation during pregnancy, and but of course a large multi-center trial that was once presented in 1994 did not find any significant difference. But I think the important thing was they should have selected the subjects appropriately. I think there are subjects, there are women who develop hypertension during pregnancy. Because of low calcium, they will benefit. But there are women who will not benefit as well. They need to find some other parameter. And in this regard, I think they need to look at the renin activity, because we know that those women who have low renin activity will probably benefit from calcium supplementation. Now, we had wanted to do this study, but the ethics committee did not agree when I was in USM for calcium supplementation to the mother, so we did not follow that study further. But it is something that we may want to explore sometime again. But whilst we were doing that, we also started looking at the placenta, and we started looking at some placental factors that may be involved. As you all know that the placenta is the major factor that actually is involved in preeclampsia. I'm not saying placenta is the only one. There are some maternal factors as well. But at the end of the day, we know that if you remove the placenta, once the placenta is removed, most of the blood pressure and all the markers of endothelial activation, the proteinuria soon disappears. Although we also know that there are women who develop hypertension after childbirth as well, immediately after childbirth. But nevertheless, overall, the view is still correct that placenta may be one of the major factors that is involved in pregnancy-induced hypertension. So we were measuring some of these vasoactive peptides in the placenta of women with preeclampsia. And what we found that the level of endothelin was significantly higher in the fetoplacental tissues of women with preeclampsia. We also measured the renin concentrations, activities in the placenta. And you'll be surprised, we found that the levels of prorenin were significantly, very significantly higher in placenta of women with preeclampsia. Now, what is the role of prorenin there? We do not know. But what we know is that the level of the concentration of prorenin is highest in the gestational sac, and then it decreases as gestation and pregnancy progresses. But it appears that women with preeclampsia, the level does not fall as much as it does in normal pregnant women. So this is something that we have not, although we have published this work, but we have not been able to do further work on that yet. But that is something worth looking at now that we have more tools and better understanding of the prorenin. We also looked at the tissue kallikrein and kininogen concentrations in the fetoplacental tissues. These were also different, and this is the vasodilator, and they were significantly lower in women with preeclampsia, right? The placenta of women with preeclampsia. Now, we wondered, what about leptin? Now, this is something very interesting. We had these tissues from our previous studies. They were all frozen, and we had just completed these studies, and there was a little incentive grant that was made available by USM to the scientific officers and senior technologists in the faculty or in the university. So what we did was it was a maximum of 3,000 ringgit for a simple study. So I discussed with one of our technologists, our senior scientist, and she was interested and wanted to apply for it. So we said, all right, we have the tissues. Why don't we just measure the level of leptin in these tissues? So she got about 3,000 ringgit and we bought this leptin kit for about 2,700 ringgit and we measured. And what did we find? We found that the leptin levels were significantly higher in the fetoplacental tissues from women with preeclampsia. And this paper was published in a Q2 journal. It costed us about 2,700 ringgit. Then we asked ourselves a question again. Is there a role for leptin in pregnancy-related hypertension then? If it is high, and by then we knew that there were a couple of studies that had shown that the serum leptin concentration was higher in women with preeclampsia. But these women were also overweight and obese. So some people thought this higher serum levels might be secondary to their larger amount of adipose tissue. But seeing them in the placenta now gave us a different idea. They said, well, it may not just be a passive bystander. It might have some role. Does it have a role? This is what we asked ourselves. So what we did was we designed a study, and I had moved to UITM then, and we applied for a short-term grant, Dana Kacamarlangan, and it was approved. And we did this study on rats. Again, we used our Sprague Dolly rats, normotensive, normal weight female rats, and we actually mated them with males, normotensive males, and we started giving them leptin from day 1 of pregnancy to see what happens. And what did we find? Now, here we have 3 groups of rats. We have one group. This one is the normal non-pregnant rat over the period of, now the duration of pregnancy in a rat is about 22 to 23 days. We euthanize them on day 20 so that we can collect the blood and do all the necessary investigations. So this is the non-pregnant rat. This is the normal pregnant rat that was not given leptin, and this is the group of rat that was given leptin. And look what has happened to the blood pressure. It has actually increased. So those rats that were given leptin, during pregnancy, the blood pressure, maternal blood pressure, increased over the duration of pregnancy. We also found, I've not included the data here, there was some impact on the litter as well. The size and the litter weight was slightly lower in those dams or those female rats, pregnant rats that were given leptin. In addition to that, apart from blood pressure, we know that women with preeclampsia also have proteinuria. They also have evidence of endothelial activation. So we actually looked at the protein excretion in these animals. And this is the normal non-pregnant rat. All right, there was no change over the 20-day period. This is the normal pregnant rat that was not given leptin, but just given saline. And look at what happened to the protein. Of course, during pregnancy, we all know that urinary protein excretion increases during pregnancy. But more importantly for us, look at those animals that were given leptin during pregnancy. The urinary excretion, protein excretion, was significantly higher. Very similar to what you see in the human. Look at the urinary albumin excretion, the same pattern. Urinary creatinine clearance decreased in rats that were given leptin. We looked at the markers of endothelial activation. Look at what happened. There's increase in endothelin 1. And we had already reported in placenta that it increases. And we now know that the serum level of endothelin 1 is also raised in women with preeclampsia. Look at serum Is Leptin. These are all markers of endothelial activation, ICAM. But I want to draw your attention to this particular thing, which has not been done before. It has never been reported before. And what we found was that the ACE2 activity was significantly lower compared to that in the normal pregnant rats. It was not different from the control rats, but it was significantly lower. Normally, now we know that the ACE2 activity increases during pregnancy. Now, just for the benefit of those who may not know much or perhaps are not very familiar with ACE2, ACE2 is an enzyme that was discovered some 20 odd years ago, and recently it has been spoken a lot about because the COVID virus actually binds to this enzyme, and that's how it gets into the cells through this enzyme. Now, ACE2 actually is an enzyme that counterbalances the effects of ACE, and we know the conventional angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. Now, angiotensin 2, of course, causes vasoconstriction, increases peripheral resistance, increases blood pressure. It also increases the secretion of aldosterone and increases salt retention. But what ACE2 does is it actually breaks down angiotensin 2 to angiotensin 1-7. Angiotensin 1-7 then binds to a receptor, the mitochondrial assembly receptor, or abbreviated as MAS receptor. By binding to the MAS receptor, it causes vasodilatation. So in other words, it actually counteracts the effects of angiotensin 2. And so there is this counterbalance system. So this is the other arm of the renin-angiotensin aldosterone system. Now, this is the first time it has been reported in this situation, and the first time, of course, it was reported and the effect of leptin on this particular enzyme activity. We have subsequently confirmed that as well, because there are other studies where we have measured this, and we find that leptin actually decreases the expression of ACE2. Yep. And these results, of course, have been published in a lot of journals, all of these. And what we also found was, now, we thought if ACE2 was involved, now this is where the confusion is going to come a little later on, and something that we'll have to now try to answer, and it'll open up more areas for research, of course. Now, if ACE2 activity was suppressed by leptin, what if we actually increase the ACE2 activity? Could we prevent the effects of leptin? And there is a drug called xanthanone. I don't think it is used clinically that often, but it is known to actually upregulate ACE2. It activates ACE2. So we repeated the study. What we did was we gave leptin to the rats, and at the same time we gave xanthanone. And what we found was actually it prevented the effects of leptin. Now that's very interesting. It prevented the increase in blood pressure caused by leptin. It prevented the proteinuria as well during pregnancy. Now it's confusing a lot of things now, but this is what we found. We have published that in Reproductive Toxicology, which is a Q1 journal, and it's opened up sort of interesting avenues for us as well for further study. Now we went on again. We repeated the study. This time we also found that not only xanthanone, but we also resibuforgenin prevented the leptin-induced effects on blood pressure and markers of endothelial activation during pregnancy. Now, what is resibuforgenin? Now, there is evidence that marinobuforgenin, this is a factor produced, it's a sodium transport inhibitory factor. It gets its name from a bullfrog, a South American bullfrog, because it produces this poison. So that's how it got its name. But there is evidence that women who develop preeclampsia, the level of marinobuforgenin is actually increased. So it perhaps is also inhibiting the sodium transport in cells and might be responsible for the raised blood pressure to some extent. And resibuforgenin is an analog of this, and it blocks the actions of marinobuforgenin. So when we give resibuforgenin, we found that it prevented the effects of leptin. Now the question is, what is the relationship between xanthanone and marinobuforgenin and all this? Now this is something we have to solve someday, but that is what the situation is like at the moment. We also went on to just look if leptin would increase blood pressure in non-pregnant animals. And for this, we actually injected leptin into rats for a period of 14 days. and see what has happened. We have 4 groups here. We have the control that was not given any leptin. It's normal saline. The blood pressure increased slightly. This perhaps is because of the age-related increase that we see in rats. And 14 days or 2 weeks is quite a long life quite long in a rat's life. We also found that rats that were exercised, you see what's happened? The blood pressure remained low. Those that were given leptin, the blood pressure increased. Those rats that were given leptin and exercise, the blood pressure was very similar to the ones that were exercised. What he told us was that exercise also prevents leptin-induced increases in blood pressure. Yeah, so we have shown over the number of studies over the years, beginning with the studies on reproductive system in the humans, we found that melatonin prevented the effects. We do not know, we have not investigated the effect of melatonin on blood pressure in leptin-induced animals, but from our previous studies, we know that melatonin actually decreases blood pressure in rats, in spontaneously hypertensive rats. I've done quite a bit of work on that as well. And we know that melatonin decreases that. Not only that, when we gave melatonin to pregnant animals, spontaneously hypertensive rats, what we found was it actually delayed the development of high blood pressure in the offspring, even when we did not give them melatonin after, but it delayed the development of hypertension. So melatonin is known to have some effects on high blood pressure. But more importantly here, what we have found is that exercise decreases or prevents the action of leptin. Again, it goes to show the importance of exercise. And we looked at the level of endothelin-1. Again, the endothelin-1 was elevated. These are non-pregnant animals. The level was elevated in leptin-treated animals, and exercise prevented that. All right. So we've published this work in a journal, one of the European journals. So what are the possible mechanisms? How does leptin increase blood pressure? There are 4 possible routes. They all 4 may be involved. One, of course, we know that leptin increases sympathetic activity. So increases sympathetic activity would perhaps increase the vasoconstriction, might increase blood pressure. It also causes endothelial activation, and in this regard, we know it increases endothelin-1. And endothelin-1 is a vasoconstrictor. So it's possible that there is endothelial activation, and that activation is increasing blood pressure, causing vasoconstriction, increasing total peripheral resistance, and increasing blood pressure. It's also possible that the effect of leptin on blood pressure may be through decreased ACE2 levels. All right. I mean, we tend to measure renin activity in hypertension. Of course, it's very difficult to measure, and it's very rarely done. But I think when we look at that, again, we need to actually also look at ACE2 at the same time. It's possible that a lot of hypertensives who develop high blood pressure may have low ACE2 level and activity. And of course, leptin might also be altering salt and water balance, because although I did not present the results here, we found, and it's known that leptin actually stimulates tubular reabsorption of sodium. So it's possible that leptin might also be increasing the extracellular fluid volume through sodium retention. So these are some of the possible mechanisms through which leptin might be increasing blood pressure. Now, we are also, apart from this, we've also looked at the effect of leptin on blood glucose. And we know that, there are lots of obese individuals have, dysregulation of glucose, and diabetes is more prevalent in obese individuals. So for this, what we did was we actually injected leptin into rat, daily injections of leptin for 6 weeks. And after that, we actually challenged them with a glucose load, an intravenous glucose load. And we measured glucose afterwards. All right. So we infused glucose over 5 minutes. So the control and the leptin-treated rats had glucose, same amount of glucose infused over the same duration of time. And we measured the plasma glucose level. And this is the control animal. And these are the animals that were given leptin. And look at the glucose concentration. So slightly higher than that in the normals. In other words, glucose clearance was not as good as that in the normal rats. Look at the insulin level. This is that in the control. Look at the insulin level. The insulin level was some 4-fold higher in leptin-treated rats. So in other words, there was some sort of decrease in insulin sensitivity. And we calculated the insulin sensitivity, and it was lower in leptin-treated rats. We have not investigated this further, but it suggests to us that we must also consider leptin and its role in glucose dysregulation in the obese. Now, apart from this, we published this work a few years ago. We also looked at leptin and cancer. Now, there is a lot of evidence in the literature that the prevalence of some types of cancer is higher in obese individuals. And we wondered if leptin had any role in that. So we investigated the effect of chronic leptin administration on gastric adenoma in the rats. And for this, what we did was we injected 60 micrograms per kilogram of leptin daily, and this was given for 40 weeks. 40 weeks is a long time in a rat's life. So we injected daily for 40 weeks and to see what happens. And what we found was very interesting. We found that, and then we looked at the expression. We did microarray analysis, we did RT-PCR to look at the effect of leptin on some of these tumorigenic genes. And what we found was leptin induces the expression of some of the tumorigenic genes in the gastric mucosa of the male Sprague-Dawley rats. So could it be that the higher levels of leptin might be responsible in the higher prevalence of cancer in obese individuals? Not only that, what we also found was that leptin also enhanced the tumorigenic effects of a carcinogen MNNG. This is a common carcinogen that is used in research studies, that is used to induce gastric adenoma cancer in rats. And we found the level of this was that leptin actually enhanced the activity of this carcinogen. I mean, how do we interpret that? I mean, we are exposed to a lot of carcinogens, it seems, in our diet, in the atmosphere and what have you. And is it possible that with higher leptin levels and these carcinogens in the diet, these could be actually causing that higher prevalence of cancer in obese individuals? Something to think about, something to work on, something to research on. And what else we noticed was in these rats, we have not published that study yet. We wanted to do a little more work. That in these rats that were given leptin, they also had higher evidence of lung cancer. There were growths in their lungs. We could not tell at this point. We have not really looked at to see whether these are metastases from the stomach or these are the primary sites. So it's possible that leptin also increases the prevalence of pulmonary cancer. So that is something that we need to look at. Now, if I may recap all this, what we have done so far, and I'm coming to an end my talk. We know that leptin reduces sperm count, increases fraction of abnormal sperm. And we also now know that the most probable mechanism is oxidative stress. And the most likely pathway is the PI3K pathway, although we know it's not the AMPK, but we still need to exclude the other pathways. But the most likely pathway is the PI3K. And we know these effects are reversible. What else do we do? How do we now utilize this information? How do we translate this information for clinical use? And what else is there we need to study? What are the effects of leptin? How does leptin get over the testis barrier? Does it get over the blood testis barrier? How does it do it? Does it affect the membrane proteins? What does it do? What else does it do? So there are lots of questions that we need to answer yet. I mean, these are just indications of the role of leptin, but before we can fully understand that, there are lots of studies that are required. Now we know that leptin also increases blood pressure and proteinuria during pregnancy in the rat. Something that is, some of the findings are very similar to those that we observe in women with preeclampsia. And we know that these effects are prevented by xanthanone, which is an ACE2 activator. And we also know that these effects are prevented by resibufogenin. Now, as I mentioned earlier, how do we relate these? How can we come to a common point? Where is it? So all these questions still require a lot of research work to answer. So this is something that has confused a lot, but we need to really look at that. And we know that leptin also increases blood pressure in non-pregnant rats, and it might involve a number of mechanisms, and we know it is prevented by exercise. So what else? What else do we need to know? We need to study this further. Yes, I think my own personal opinion is exercise should be prescribed for everybody from very young itself, and you can prevent a lot of ailments. Can exercise be a good treatment modality? What else do we need to do in individuals who are already hypertensive? So certain things, we need to look at. What are the parameters should be? What are the investigations can we do? We know that leptin also causes dysregulation of glucose metabolism. How does it do it? Insulin levels are raised in leptin-treated rats. Now, insulin, of course, is a very important hormone. We need it for glucose metabolism, but we also know that insulin is very lipogenic. And so what is the role of leptin in atherosclerosis, for example? I think this is something which we need to look at. I think personally, I am of the opinion that leptin actually increases the rate of atherosclerosis. So something that we can work on and look at it. And also, what is the role of adiponectin here? Does adiponectin counter the effects of leptin? Is there any relationship between? Because we know that in the obese, as leptin levels increase, the adiponectin levels decrease. So what is that, the relationship between these two? Of course, just our recent studies, they are preliminary. They are the first studies of its kind. Nobody else is. Although there is a lot of evidence in the literature showing that leptin increases proliferation of cancer cells, but whether it actually causes, it's also tumorogenic has not been reported before. Ours was the first study that has actually shown that leptin might be tumorogenic. And also it enhances the effect of carcinogens. So these have to be looked in further as to how does it do it? What are the mechanisms involved? So these are some of the questions that we need to answer. And we also know that, as I started off, about obesity, that obesity is actually related. There are lots of obesity-related diseases. Yeah, and some of them now appear to involve leptin. All these involving leptin is something that we need to look into. My own hypothesis, my belief is that leptin could be the link between obesity and obesity-related disorders. Now, I've presented quite a bit of work that we have done. Of course, as usual, it's not all my work. There is always a team. There are a lot of people who contribute, and it's only fair that I think they all be acknowledged here, and their names are here. We have an adipokine interest group that we started here over the years, and these are the members who have been involved in that group. From time to time, some of them have left us and they have moved on. Some of them are still here. And these are all the postgraduate students. All the work that I have presented here was done by these postgraduate students. And my thank you to them, actually. And thank you all for your attention. Thank you.
Thank you so much, Prof Harpinder, for a very interesting presentation. We learned so much from your presentation regarding leptin, and obviously you have a very, very experienced work on leptin.
Thank you.
I think it's a very fast growth of knowledge. You've been 11 years, but you've done lots of work on leptin, and it's very, very exemplary, Prof. We have one question from the audience. If we want to bring leptin into the clinical studies, how and where do we start, Prof? Any suggestion?
Obviously, I think in clinical studies, of course, if there are leptin deficient humans and all that, you will actually treat them with leptin. But most of these obese humans are not leptin deficient. If anything, they have excess leptin in them. Now, we cannot give them leptin. What we have to aim for is how we can actually minimize the adverse effects of leptin. I think that should be the target in most instances. Now, that is going to be a challenge. The reason being because we know, like so many other endocrine hormones, for example, leptin is required for normal physiological functions. Now, if we were to block the effects of leptin, and by the way, there aren't that many blockers available yet at the moment. There may be a few on trial or under development. Some antibodies, for example, against the leptin receptor, the leptin, but we cannot give them just like that because there is a possibility they will cause a disruption in the normal function. So the challenge now here is, at the moment, is how we can minimize the adverse effects of leptin. Now, in our studies, we have shown that I think we may have to look at the role of antioxidants. All right, we may have to use in those individuals who have higher leptin levels. We may want to think about using some antioxidants in them, giving them antioxidants. We may want to think of the role of physical activity, exercise, because we know that exercise, in fact, prevents the effect of leptin. So these are some of the things that we need to look at. And so really, I think in short, at the moment, we don't have any leptin antagonist that can be used clinically. Leptin supplementation is only for those who are leptin deficient. Those who are obese, there are studies, there are people in animals, for example, they have actually given them more leptin because their leptin sensitivity is low. They say if we increase the leptin level, they might reduce weight. Yes, the weight comes down a little bit, but the consequence of that is that those people go on to develop raised blood pressure and other complications of leptin. So we want to prevent that, because in most obese individuals, the decreased leptin sensitivity is at the level of the hypothalamus, but the periphery is still intact. And there are lots of receptors in the periphery, and those high leptin levels in the periphery are the ones that are actually causing the effect. All right. So really the challenge now is how we prevent the adverse effects of leptin. Right. So this is as far as I know that we can do. Clinically we have to think about leptin, and we have to think about ways how we can minimize the impact of leptin. At the moment, we don't have any particular drugs or agents that could actually prevent or antagonize the action of leptin.
Thank you, Prof. There's another question I think you did answer indirectly from the audience. Hi, Prof. Can leptin-related hypertension be reversed with exercise rather than prevented?
Yes, I think from our animal studies, it clearly showed actually that you exercise, it reduces it prevents the effects of leptin. All right. Now, because we started off giving leptin and exercise at the same time. There was one group of rats, if you recall my slides there, and it prevented the effects. In fact, not only that, as you would see, that it prevented the age-related increase in blood pressure. In fact, what exercise does, it has been known for a long time anyway, that regular physical activity prevents an age-related increase in blood pressure. And it also decreases blood pressure in hypertensives. So those individuals who are obese and where you suspect they may have high leptin levels, yes, encourage them to exercise regularly. You may require less antihypertensives. Yeah.
A very basic question from myself. Prof, I mean, like the leptin is like being expressed in our whole body, right? I just wonder, is there any mechanism that actually regulate the leptin expression in terms of gene stage?
I think this is a very, very important, interesting question, Swaiza, because now leptin, most of the leptin that you find in circulation is produced by adipose tissue. All right. Although small amounts, as I mentioned just now, is produced in the stomach, is produced in the mammary gland, is produced, and quite a lot is produced in the placenta. But of course, that is during pregnancy. But in the normal situation, most of the leptin is produced by the adipose tissue. Now, the challenge here is how can we, just like in the thyroid we do, how can we stop the adipose tissue from producing leptin? I think if we can suppress leptin secretion by the adipose tissue, so we can have a obese individual but lower leptin levels, and he may not suffer from, he can still enjoy his food, he can enjoy life, he can live to eat instead of living to eat, but he can sort of live to eat rather than eat to live. But that is the challenge. I think that is something which is worth thinking about. How can we regulate or control leptin secretion? Because leptin is produced constitutively by the adipose tissue. All right, so it is being produced, as I mentioned earlier, the amount of adipose tissue there is, the higher the leptin. There is a very good correlation. The bigger the fat cell, more leptin it produces. Now, how can, because it just produces, it does not store, all right? It just produces and it releases as it goes. There is a slight variation, all right? Immediately after a meal, a few hours after a meal, leptin secretion increases. But I think that is one area that we need to look at. How can we reduce adipocyte leptin production? And if we can do that and bring the leptin levels to normal level, I think we will probably prevent a lot of obesity-related diseases.
Thank you, Prof. The other thing, obviously, we are now in what we call PKPB, and one of the great issue, I guess, is the COVID-19. And most of the patients who actually have comorbidity is the patient who have all these risk factors, Prof, like diabetes, hypertension, and unfortunately, one of it is obesity.
Yes.
So, yeah. So one of the recent articles I say they actually have looked to this patient and they actually find out the obesity is one of the confounding factors for COVID-19 outcomes. So what do you think, Prof, the role of leptin? I think there are clues there because we did mention about ACE2 just now, right? Yeah.
I think obesity or people who are overweight, obese, actually have what is known as low level of inflammation in their body. In fact, obesity is a low inflammatory state. So in other words, even under normal circumstances, an obese person may seem as if he is happy and healthy and, but actually he's not well. The endothelium and all that is activated and there is low level inflammation. And I think that particular inflammation may be what is making them more susceptible to COVID infection. Because even in diabetics, for example, if the sugar is not well controlled, they have a lot of endothelial dysfunction and activation. So it's possible, and hypertensives also have that problem. So it may be that, the endothelium, when I first read about COVID-19 using the ACE2 enzymes, as its vehicle to get into the cell, and I thought to myself, oh, then obese individuals perhaps should be protected in a way. Because there is leptin and we know leptin decreases the expression of ACE2. So they express less ACE2 and there'll be less ACE2 there. But it didn't seem that way. So it's not quite the ACE2, but it's the inflammation, I think, that is perhaps responsible for it. Yeah.
Thank you so much, Prof. I think that's quite a good point to start. I mean, like, even if you want to do something regarding COVID-19, I mean, like, it might be something that can help out because we kind of mysteries and having a little bit clues, right? So, yes. Because leptin, if we have a look, as mentioned by Prof, it does involve in hyperglycemia, hypertension. It's like we say it's on top of all the comorbidity that is related to. Yeah.
I personally think it is a link between obesity and all obesity-related disorders.
Yeah.
So we have shown in at least 4 or 5 of them that it might be involved. There are a lot more to do. Yes.
Exactly. It might be the key to the answer of the mysteries, Prof.
Yeah.
With all the related. So I say thank you so much, Prof. We, the team, Faculty of Medicine, really appreciate your effort and also all the knowledge that you have shared to us today. We learned so much from this presentation. I think personally, I also learned so much about leptin. Hopefully, everybody, the audience here also can gain the knowledge and also might open some opportunity to do research, especially in leptin, either in preclinical or clinical area. So thank you so much, Prof. And thank you so much to the audience. Do you have anything to say to the audience, Prof, before we complete our presentation here, our session?
Yep. Well, thank you very much for being here today. But if you have any ideas or if you have any interest, research that you want to undertake in leptin or you have some ideas, please feel free to contact me. I will be happy to share my thoughts with yourselves and perhaps even prepare a proposal to look at the effects of leptin in disease. Yeah.
Thank you so much, Prof. And thank you, everybody. Okay, so with that, I say goodbye and thanks again. Okay,
Thank you.
bye-bye.
Thank
Thank
you.
you,
Take
Prof.
care.
Thank
Thank you.
you.
Take
Take
care. God
care.
bless. Take care.
Bye.